Hello, everyone. Can you see my screen? I can't see the chat. Okay, I will take that as a yes. I will go until somebody stops me. Okay, we are all good. So thank you everybody for having me here to talk at this symposium. It's wonderful to be here in this kind of hybrid space. Let me move things around. Okay, wonderful. So today, my name is Isabel Johnson. I've been working in Dr. Fei-Wei Lee's lab. We are looking at hornworts, and today I'm going to present on my summer research that has to do with the investigation of a hornwort insertion line with rhizoid-specific GFP expression. So, why are hornworts something to study? Well, the model species Anthoceros agrestis was just found about 10 years ago. Hornworts are a type of bryophyte, so they're sisters to liverworts and mosses, and they're also sisters to all other vascular land plants. They share a common ancestor with vascular land plants about 500 million years ago. So it kind of acts as a keystone species between algal ancestors of plants to the vascular plants that we see today. Other cool things about hornworts is they have cyanobacteria symbiosis. So they have a symbiosis relationship with bacteria that fixes atmospheric nitrogen into the soil so that plants can uptake them. The hornworts also have pyronoid structures, which are only found in other algae, and no other land plants have them. Pyronoid structures are carbon concentrating mechanisms, and they increase the photosynthetic efficiency of rubisco. So it sequesters more carbon than the chloroplasts in regular land plants. Both of these characteristics are very, very beneficial to the hornworts, and they would be incredibly beneficial if we were able to implement them in crop plants. So understanding more about hornworts and understanding the genomes of hornworts and doing a lot more preliminary investigations of hornworts to understand these characteristics and implement them in crop plants would be very, very beneficial. So one of the preliminary experiments that we did this summer is I focused on the rhizoids of these hornworts. Here you see the gametophyte, and the rhizoids are the little hairs that are coming off of this little guy. They are one cell long, so they're one cell. They have one cell wide as well. So they they're used to anchor the hornworts to the land, and they also are used to uptake nutrients. If you want to think of them as root hairs, that's also kind of what they are. It's a good way to think about them. So why are we interested in the rhizoids of hornworts? Well, this is a plasmid that was bombarded into a hornwort using a gene gun. So the plasmid was bombarded into the hornwort's genome, so it should be, if it was successful, it should be expressed in the hornwort genome. The plasmid contains a fluorescent gene called GFP, where it fluoresces under UV light when expressed. So if the bombardment was successful, we would expect the entire hornwort to fluoresce green. This is an image of the BE3 strain, which I was studying this summer. As you can see, it was bombarded with the GFP, but the only thing that's glowing green are the rhizoids. It's not the entire hornwort, which is not expected. It is very, very cool. So we don't really know why this is happening. So we did investigations to kind of see, well, why is the GFP only expressing in the rhizoids of this hornwort that was bombarded with this plasmid? So the Illumina data was gathered here, if you can see, and then you can see it fragmented a lot. The Illumina data does a lot more shorter reads. So there's a lot more fragmentation in the GFP, in the plasmid insert that you can see here. So we hypothesized that there is a rhizoid specific promoter that's acting as the promoter for GFP, because if you can see here, there is no elongation factor near the GFP. So we think that one gene that is specific to rhizoids is acting as the GFP promoter. To kind of investigate this, we want to see if the genes around the GFP insert are in fact expressed in the rhizoids. So what we wanted to do was harvest the rhizoid tissue from the hornwort, and we did that by growing the hornwort on 2% sucrose media to make them generate a lot more rhizoids than normal. You can see this guy is a very hairy hornwort. So when it comes to actually like getting the rhizoids off, we had two methods. They were both based on Middle Eastern cuisine. One of them was the Kebab method, where we stuck little hornworts on a skewer. We dipped them in liquid nitrogen, and then we twirled them around in a test tube to make the rhizoids very brittle and kind of fall off. So that didn't really work as well. So we moved to the shawarma method, where we shave the rhizoids off the hornworts, much like you shave the meat off of a shawarma. I don't know why we based our methods on Middle Eastern practices. We must have been hungry. So this is the kind of kebab that you can see on the left that we did with the hornworts that we dipped in liquid nitrogen, twirled them around, and you can see the shawarma method on the right. The shawarma worked a lot better, so that's what we stuck with. So we did get the RNA from the rhizoids to do a reverse transcription PCR. This is the gel of the cDNA that we gathered. On the left is kind of our positive control, which has the tissues from all the tissues from the hornwort, so the rhizoids, the gametophyte, and the sporophyte. The genes that we looked at were 98, 99, and 101. All of those genes are around the GFP inserts, and they were kind of candidates of being the promoter for the GFP. So as we can see, the RNA from the rhizoids that is now turned to cDNA for the 99 and 98 were present. If you want to look at the red arrows, those are pointing to the 98 and the 99 in the rhizoids. Around 100 kilobase pairs. 101 was a little bit of an anomaly. We think that there was something wrong with the primers, which made 101 not absent in the full hornwort and the rhizoids. So we ran out of time to kind of test that. So so we still really don't know what the insert looks like at this point. So what we wanted to do is get a little bit longer reads of the DNA. So we did nanopore sequencing. And what we did here was a little groundbreaking. We did selective sequencing using reed-fish adaptive sequencing, which has only been done twice in published papers. We got it from Payne et al., which was very cool. So basically what happens regularly in Nanopore is the DNA is sequenced through the pore, and it's not really specific to any type of DNA, it's just any DNA available. What we did is we programmed the computer to recognize our target DNA, which was the Contag 83 that had our GFP insert on it. So we wanted to target that specific contag in the DNA. So we told the computer to recognize it. And if our target DNA that had the contag 83 went into the pore and was sequenced, and the sequence was recognized, it was continued to be sequenced. But if the DNA was not recognized to be contag 83, the DNA was actually ejected from the pore to leave the pore open for other DNA to come in. So this leads to more reads of our DNA that we wanted, the contag 83. It also led to longer reads of the DNA. This is a graph showing that was successful. The reed-fish data was successful. The red line represents our contag 83, the segment of DNA that we were looking for that has our GFP insert in it. As you can see, the depth for that one is a lot higher than the depth for the average of all the other contags, which is very exciting that we were able to do this successfully. Here is an overview of the GFP insert that we got from the DNA sequencing with Nanopore. It was assembled using Fly. It has 680,000 base pairs. The insert is 6,800 680,000 base pairs, which is pretty big. And it's in between the gene 100 and 102. Here's a little bit of a closer look on all the GFP inserts that were actually in the plasmid insert in the DNA. As you can see, there is a lot of GFP present, and there's also a lot of promoter elongation factors as well. So a bunch of the plasmid landed in the DNA. With this amount of GFP in the DNA, with all its elongation factors around it, we would expect for the entire hornwort to, in fact, glow, which kind of disproves our other theory. You know, there's not any promoters acting on the GFP, it has the promoters on its own. So this begs a question, you know, why are our rhizoids still glowing? Why isn't the entire hornwort glowing? So we needed to come up with a new hypothesis. Since there are so many GFP present in the plasmid insert, there must be we think that there's some type of epigenetic factor responsible for the silencing of the GFP gene in every other tissue but the rhizoids. So we think that there could be some histone acetylation or DNA methylation that could be silencing the GFP in other tissues but the rhizoids. To kind of do preliminary analysis of if we're on the right track, we did a pairwise comparison of the genome of Anthracis agrestis, our model species, with the genomes of 6 other hornwort species. So we looked at syntenic blocks that were shared between every single species, and we looked at the specific genes that surrounded the GFP insert, and those genes actually landed in one of the biggest syntenic blocks of the hornwort genome of this pairwise comparison that we found. So I named this block Steve because GFP insert syntenic block was a little too hard to type when I was coding. So you can see Steve here in the red of this violin plot. The y-axis represents the number of genes that are found in the syntenic blocks, and the x-axis is Anthracis agrestis compared to the 6 other hornwort genomes. So if you look at the red dot that represents Steve, you can see that he is an outlier, and this proves that the genes that the GFP landed around, or the genes around the GFP insert are are contained in this syntenic block that is very, very big. This is consistent with our hypothesis of epigenetic factors, mainly because if these genes are easier to control, we would expect for more syntenic relationship to exist between all hornworts. Okay, we also looked at the GO enrichment factors with this specific syntenic block over a bunch of hornwort genomes, and we found that the DNA binding transcription factor activity was enriched in all in this syntenic block in all of the hornwort genomes. What does this mean? This means that these genes that are in the syntenic block aren't just put together haphazardly. There's some type of organization. They are not just together randomly, but they are assorted to have one specific function. This might be consistent with our epigenetic hypothesis. You know, when having this GO factor be enriched, we would expect maybe some epigenetic factors to control this. We ran out of time to do like any specific looking at methylation or acetylation of histones, but further analysis can be done to kind of try and see if there's any methylation of the DNA or histone regulation going on. We did do a preliminary study using TSA as a drug to make the histone, to deacylate the histones of the hornworts to try and get GFP to express everywhere, but that didn't seem to do anything, but we can't really rule that out. More detailed work is needed. But we can also try to demethylate histone or demethylate the DNA in the hornworts in the future. And another study is that we can also see if the genes around the GFP insert are expressed in other tissues other than the rhizoids. So that is for the research that we can do, but that is the end of my presentation. I would like to thank everybody at Boyce Thompson Institute, NSF, USDA. I would like to thank everyone in Lee Lab. You helped me all very, very much as a team. Thank you to Fei Wei Li, our fearless PI, and also Peter Schaffrin, who taught me basically everything I know about bioinformatics. So thank you.
Questions here?
So I cannot hear any questions if any questions are going on. Sorry. I can't hear at all. I'm so sorry. Okay, now I'm good. I can hear.
Okay, try again.
I'm sorry, I can't hear what they're saying.
Hi, great talk. Did you see evidence of GFP integrating into some of the other contigs that you sequenced in your nanopore sequencing, like the untargeted contigs?
Yeah, so actually, let me go back to the slide. We didn't see any other, let me go back to it. Where is it here? Okay, yeah, we actually didn't see any other GFP inserts or the plasmid insert at all in any other place. I don't know if you can see my cursor, but the GFP only really, or the plasmid insert only really landed in this portion. It wasn't found in any other portion of the contig, so this is like kind of the only place that it could be expressed, but it's a good question. Thank you.